Okay, welcome everybody. Today's talk will deal about experimental determination of biological macromolecules. I won't talk about X-ray crystallography because I did it already so many times. Today I tell more about electron microscopy, a method that is quickly getting pace now in structural biology, as can be seen from the increase of structures in the protein database. First of all, as you may note, here, cryo-electron microscopy today is able to resolve structures not only of large entities like viruses, as it has been used before, but also of ribosomes since about 10-15 years, and nowadays getting smaller, to enzymes like this one with about 100 kilodalton, or even hemoglobin with about 60 kilodalton. So before we go to this kind of techniques for getting these kind of structures, something to mention about electron microscopes. Electron microscopes have been invented in the 20s. Please remember where the Fernseher, the television set, has been developed in Berlin, mostly in Germany. And one of the side projects at the Technical University of Berlin was use the same technology. That means getting electron beams, focused and reshaped by magnetic fields to build a microscope with this. And it was his PhD project of Ernst Ruska that he did and succeeded, with Max Noll in 1931 to get the first electron microscope up and ready. And that one, the first one, was already getting to magnification that were far exceeding what you could get with an optical microscope. That's the reason why it was called the Übermikroskop. Like the Übermensch is using his Übermikroskop in 1933. Very strange number. Okay, so when you consider an electron microscope, it is pretty analogous to a usual light microscope. What do you need? You need a source for the radiation, that's the electron gun. The electrons will be then shaped, here and accelerated by the anode, onto which they are accelerated from. The voltage, between the electron gun and the anode determines the kind of class of microscope. Usual microscope nowadays have acceleration voltages, of about 120 kilovolt to up to about 300 kilovolt, maybe even some higher with some very highly dedicated ones. So this electron beam that is emerging here is getting here into a condenser, like in the microscope, that you get a uniform beam by shaping it. These are like lenses. These are magnetic lenses, Then you get to the sample itself, here shown as red dot. And finally, you need, of course, some objective, like here, like in the microscope objective, and some additional lenses, and projecting the image here on a projection screen. Usually nowadays, of course, an electronic CCD camera that is getting the image. So that is the general point. What's different? So the main difference to a normal light microscope is that electron beams migrate, of course, only in vacuum, in fine vacuum. So all the kind of measurements have to be done in fine vacuum. That means, this is, of course, in contrast to the requirement of biological macromolecules to be an aqueous solution. So everything done here has to do with sample preparation. We get later on this. So here again, the comparison also in German, Hier Strahlungsquelle, Objektiv, Okular, Projektorlinsen, Kondensorlinsen, pretty the same stuff. But you also note, the wavelength of our electrons is much, much, much shorter, than the wavelength of the light. Sorry for this, I'm not at home. Much, much, much shorter. The resolution you can get, here it's estimated with about 2 angstrom. But that is not absolutely true. The physical resolution is much higher, actually, because of the very short wavelengths the electrons do have. So it's probably in the area of an angstrom. And some people, in material chemistry and material sciences use actually atomic resolution imaging on surfaces by different methods like that. Another difference is the maximal sample thickness. So when you are in a light microscope, you have a thin film of liquid, about 0.1 millimeter or less. But because of the strong interaction between electrons and matter, this thickness has to be rather thin. Yes? So a micrometer is already a thick object. Less than a micrometer are the thicknesses of the objects you can transilluminate with an electron beam. And the other things we already mentioned. So let's go to an old method, of getting structured information from macromolecules. Yes. So the principle here is looks a bit more complicated, this slide, but I will go through it. There are two general problems you do have. First is the stability of your sample in the electron beam. So radiation damage is the main obstacle for all these kind of methods like X-ray crystallography or electron microscopy. It is different than NMR spectroscopy, where you don't know radiation damage on itself because it's not strongly interacting with matter. But here we have strong interaction with matter. We have reactions happening, inelastic scattering, that means energy deposited into the sample itself and causing its destruction. So that's point one, radiation damage. Point number two is, what is a contrast? So when you remember the density of, let's say, a protein, 1.4 gram per milliliter, it's not too different, the density, than the one from the surrounding water, 1 gram per milliliter. So the contrast, in terms of electrons per cubic angstrom, is not very large. That means we have low contrast. So in the past, how was this problem solved? It is still solved nowadays. You add your sample here, which is in buffer in a pipette tip, on some carbon grid. You add it on here. You add some heavy metal stain to this, which can comprise of some, like uranyl acetate, lead, tungsten, everything that is heavy atom-like. You let it adhere to the surface of the carbon support film. You dry it, and you use your electron microscope you have seen before. And here you see particles in this stain, this negative stain. The dark stain is allowing you to see the contrast. Here you see some regular bits. Apparently this is I could recognize the structure. It's probably GROE-L, GROE-S. Probably one can count. There's 7 dots in the line, yes, so a chaperone. So you see these kind of particles quite nicely. But please remember, what you have done here is to stain everything and to dry it, yes. So these molecules are certainly not anymore in a conformation that is biologically intact. So it's somehow, let's say, partially denatured. So you cannot get real high resolution information from this kind of electron microscopy images. So the other thing then you have to do is to use cryo-electron microscopy. There are also some other staining methods here as well, shattering methods, I will not come up with. Cryo-electron microscopy, is not using this kind of dry samples we have made here before. It's using wet samples. So the sample is pipetted to the EM grid. The excess of liquid is sucked off with some paper, and then you plunge the whole EM grid into liquid ethane, minus 160 degrees Celsius. And then you keep the grid at liquid nitrogen temperature, and then you put it into the electron microscope, and then hopefully you have enough contrast, you see the particles. Like in this case here we see some particles, not very well, but somehow around. So you have to look carefully for this to see that with the naked eye. So one thing, that was making a Nobel Prize was this step here from the EM grid to the liquid ethane. Because the colleague at the EMBL in Heidelberg, I have forgotten his name, found that in the 70s the freezing of these EM grids was not working. People tried to freeze grids in liquid nitrogen, but the samples were heavily damaged. What he found was that actually liquid nitrogen is not suitable for quick freezing because there is a short layer of gaseous nitrogen around the EM grid and actually acting as a barrier for the rapid cooling down and the vitrification process itself. So he switched to liquid ethane, which, as you can imagine, is highly inflammable. You can blow up your hut, yes. You can destroy your lab quite easily. You have to know how to use liquid ethane. But the point is, it works. With this you can vitrify the sample within a millisecond or even faster. So this is a general outline, here with staining, here without. Actually, staining is still used nowadays. For example, when you purify a protein complex and you would like to know, is this something worth to work by electron microscopy, by cryo-electron microscopy further on? What you can do is deliver the sample to the EM lab and let's make them a routine negative staining image first so that they can say this. Oh, it looks like similar like here on this picture. You have nice particles of a uniform size with almost no aggregation and no distorted structural elements. It looks pretty uniform. Go on, go for cryo-EM, go for structures, yes. So that's the reason why we use this method still nowadays, and this method takes more time, especially because it takes more time to see what's going on due to the low contrast. Something about contrast. Image contrast. I think you should have an idea that this actually is contrast, yes. The maximum of density you find on a recording device minus the minimum divided by the average density you have. And usually, when you make, for example, light microscopy, you make nice images where you see the image. It's actually where you see amplitudes, yes, amplitudes, coming up here, where actually something is thick, yes, here, here, something is thin here, yes, there, yes, you get a very strong contrast. There's a different kind of contrast. It's a so-called phase contrast. So when you consider, how the image is reconstructed by a microscope, yes, or the electron microscope, you at the end, unlike X-ray crystallography, you see a real image, here, amplitude one. You have also a layer, a layer in a plane in the microscope where actually you have the phases accumulating the information in. The phases and the amplitudes give at the end the image, the amplitudes here of the image are made by the interference of the different rays. So the phases themselves, yes, the phases themselves can be also used for detection, but the contrast here is weak, although they have more information mostly. So there are many methods, to get something from this kind of information. Phase is not lost in electron microscopy. Phases are still present. They are lost in X-ray crystallography. So you see a contrast transfer function, point spread function, defocus, all the kind of tricks and the descriptors you are using in electron microscopy. I do not go into the details about this because we would have to take more time and would also have to talk more about the mathematics behind it. But one thing, there is one way to get also the phases properly imaged, What one can use are so-called phase plates in microscopy. Here you see that in light microscopy, a normal picture, a normal more or less amplitude-like picture, pretty weak contrast. And here you see something where actually the phase information has been actually used up to make the image up, to make contrast up. It's made by a phase plate that is put in between the microscope, between the objective and the projector lens. This one has to have a precise diameter. Lambda divided by 4 phase plate is shifting the phases by a quarter of lambda, and that is then actually reconstructing by the interference to strong changes in the amplitudes here on the image. Something similar you can do also in the electron microscope. However, whereas this one here is more or less optics, these ones are very special materials, nanostructured materials, which can actually shift the phase of the electron beam by lambda quarter. This is something of the high-end engineering in electron microscopes nowadays. For example, like so-called Volta phase plates to increase the contrast and also to improve then thereby the resolution you can get. You see it before and after. You agree? After looks much better. Yes, it's also the particle, probably proteasome. That's how this structure looks like. So what about applications, of simple electron microscopy? So, when you go back in history, one of the first things people could do with microscopes to get structural information of molecules, was at that time determined by the problem that actually you need a lot of molecules, to get any kind of structural information. That is still the same case nowadays. But, the problem of the resolution at that time you had with the detectors and also the microscopes themselves. So what one could do at that time was to grow two-dimensional crystals. 2 dimensions means the crystals have the thickness of just a single molecule, and then do diffraction with the electron beam at these 2-dimensional crystals and then record the diffraction images and reconstruct by crystallographic methods the structure. One of the first membrane protein structure was the one of bacteriodopsin, solved by electron crystallography in the 70s by Richard Henderson, and coming up, at the beginning of the 90s with a full 3D structure. Here you see a projection view. We just show perpendicular into the membrane, and we just see here on the projection of a helix. So each blob here is a transmembrane helix, and we just look along its axis itself, and it's projected on a single plane. Then you can start now to count how many helices we do have. 1, 2, 3, 4, 5, 6, 7 about, yes, There are 7 helices. So first structures were made by this, with 2-dimensional crystals. The disadvantage is, here's some other structure from Werner Kühlbrandt's laboratory at the Max Planck Institute in Frankfurt, some plasma membrane protein ATPase. The disadvantage of this method is you have to grow 2-dimensional crystals, and that is very difficult because you don't see them properly. Yes, not in the electron you see them in the electron microscope when you have some, but you do not see them by your naked eye or in the light microscope. So pretty difficult to get this kind of crystals. So electron crystallography is not a method anymore of nowadays work, The reasons are quite obvious, yes, The resolution was not the best one because when you have structural disorder along the z-axis, that means along the axis where the electron beam is coming from, it was taking away your resolution. Just remember the molecule thickness, 2-dimensional crystal of 1 molecule thickness, and it has to be absolutely plain, yes. Otherwise, it is wobbly. It's a wobbly crystal, yes, which is difficult to get, The sample preparation is difficult, yes, and only a few labs actually made this at the end and were doing for this. So this method was not giving the big jump in the number of structures solved nowadays by electron microscopy. So the answer to this is single particle electron microscopy and tomography. There are several pioneers who developed these methods. For example, Wolfgang Baumeister at the Max Planck Institute of Biochemistry in Munich, he developed tomography methods. Unfortunately, the Nobel Prize gets only be awarded to 3 persons, not to 4 persons. And his other colleague, from the United States, he focused mostly on single particle analysis. Let's go on with single particle analysis, what this is. First of all, just to remember again and again, the process itself of getting something in the electron beam looks really simple, yes. You have your sample, you apply it to the EM grid, yes. It has to be diluted properly so that you have not a too high concentrated solution. You need only a few microliter. You do not need a lot of material. In the ideal case, you could say, maybe I can make a structure with 50 or 100 microgram of protein. Why not? Because it gives you quite a lot of samples, EM grids, which you can use for structural analysis, different than in X-ray crystallography. You need milligrams, or NMR spectroscopy, where you need milligrams, yes. So you do the plunge freezing, and then you have the biomolecules, here, vitrified in this aqueous thin layer, yes, through which you can then run your electron beam. So that looks pretty fancy. What is not so fancy is that the sample preparation is actually the alpha and omega of electron microscopy. Everything that affects your sample quality will give some rise to some good results in cryo-EM or bad results. So the sample has to be more or less homogeneous. It has to be the protein. Maybe when you do size exclusion chromatography and it looks like that you have a very nice, well-separated peak with your sample. When you take this peak, yes, you may find out that, for example, the molecules from the rising flank of the chromatography peak, when you take these kind of elution fractions and you put it on the cryo-EM grid, look much different than those ones in the highest portion of the peak itself or in the decreasing flank again, because the molecules are not absolutely identical. So you can check up several fractions of your size exclusion chromatography, which gives the best sample, yes, for cryo-EM. Freshness has some role. The buffer can be very important for stability of conformations. The expression system, the temperature, the mood of the experimenter, whatsoever, I don't know. There are so many aspects which may play a role from getting from some very first results in EM to really final structures. So here's something more for machine-interested people. This is a Dual 2200 with a field emission gun mounted on top. This is the tube, the high vacuum tube. This here is a typical EM grid transfer unit. Here's a liquid nitrogen dewar, so that keeps the cryo-EM grid permanently cooled at the frozen temperatures, and then you can plunge into the microscope through this kind of I forgot what Schleuse means in English, yes, a portal, yes, to get into the high vacuum of the electron microscope itself. So that is the thing. So that is a typical images you get, from a cryo-EM experiment. So where is the signal? Now the signal is where the numbers are, yes, It's not this one here. This is an artifact, yes, The signal is, and I don't know how you can see it on your computer screens. We see something there, something there, something there. So this is already an image of a membrane protein, calcium release channel, that has already done some contrast improvement by defocusation, which is one of the methods to get a better contrast, of these images. But you see, the contrast is very, very weak. So what are you doing? You are actually using computer, of course, What you're using is image recognition software of the most dedicated kind. And the imaging software is trying to find actually the blobs, in these kind of images which may be related to something of your target sample. For example, here, are the blobs where my cursor rests for some time. So these are nowadays automatically identified, and maybe you sort them out, yes, by hand, some of them, and then reconstructed and averaged here in between. And when everything goes fine, you get something like this. This one is from 1999. So it's the structure of the ATP synthase. Here is the F1 portion, where the ATP synthesis reaction is taking place from ADP and phosphate. That's the transmembrane portion here with the blobs around. That's because of the detergent that is bound to this protein. And here, so you see some color. That is actually the C subunit, that is here apparently like a disk, because it's rotation averaged. Yes, there's not a lot of structure in it. This kind of subunit is not very soft. You see some resolution here in the F1 portion, but not in the rest. So that looks like a low resolution structure. So these are the examples, from the beginning of the 2000s, how electron microscopy was used at that time. when the resolution was not high enough. Here is, for example, it's the cell puncture apparatus of a bacteriophage, T4. That is the thorn or the injection needle by which actually the bacteriophage can get through the bacterial cell wall and plunge through and get then finally injected the DNA of its own viral content, into the bacterium. It looks really like a really very penis-like structure, yes. And you see what the people have done at that time. They have made, let's say, some kind of a jigsaw by using high-resolution X-ray structures and putting them into this kind of lower-resolution cryo-electron microscopy reconstructions. So what about the data processing? I think we mentioned something about this. Here you see some roundish samples. How is it going on? First you record the images, then you identify the particles. That's done by the software. Then you do alignment of the particles, rotation and translational alignment. Then you try to make a 3D reconstruction. I come to this shortly later, why this is working, Then you get some, let's say, some structure. With this kind of 3D structure, you can try to make the alignment better, and again, and again, and again. You can also put in a 3D model if you have one of the structure, for example, of a virus, of a related virus. So what people would take, for example, so when I would have to solve the structure of SARS-CoV-19, the nucleoplasma protein core unit, and there exists some structure of SARS from 2003. Yes, it's quite obvious that you would use that kind of structure to align all the particles from this new SARS virus. Yes, because it's pretty similar, to get an optimal alignment and most optimal start point for your 3D reconstruction. And when you have done this, so it's the identification. When you have done this, you get something like this here. Yes, So this is an example. I think it's a pyruvate dehydrogenase or something like that. From some bacillus. Which is also a pretty large complex anyway. You see the 200 angstrom diameter is for a very large, highly symmetrical complex. So you wondered, why this works. Yes, so that we make images from these many, many particles and get to a final structure. First point, when you have thousands and thousands of these particles in the on the grid, yes, in this frozen liquid film, you can mostly assume that they all take random orientations relative to the electron beam. So that means, that what you do see are projections of these three-dimensional objects. Every image is a projection of the 3D object here in some kind of an orientation, but you do not know it from the beginning. The interesting point is given by the projection slide theorem. What is it saying? When you have a 3D structure and you project it now from that direction, zack, here onto that plane, and you get some projection image. It is highly related, it is identical, or let's say, it can be directly converted by Fourier transformation to the Fourier transform of the whole particle. This here is the Fourier transform of the whole particle into the reciprocal space, and then this projection corresponds to one of the central planes, one of the orientations going this way or that way, depending, on the orientation. So we went upward down, so here's the plane. Now we get this kind of slice, that corresponds to this kind of Fourier transformed structure. The funny thing about Fourier transformations is you can run them in both directions. Zack, 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 zack. So what's cute? So when you have all these kind of projections, yes, you can try to assemble your structure, all the kind of slices, all the kind of central slices, in the Fourier space, reconstructive in the Fourier space, the complete structure, which is of course the transform structure, and then do the back transformation to get the intact structure, the back projection here, to get from 2D to 3D. And that is giving you then all the structures for the single particle analysis. So it means, you collect thousands and thousands of particle images, which are projections, to be able to reconstruct, this kind of also in the Fourier space, the complete Fourier space, and do then the back transformation into the real space to get the real particle. So that is cool. That means, single particle analysis works quite nice. So why is it not always working? So one of the reasons is, one of the requirements we mentioned before is that all the particles adopt a random orientation. That is not always true. For example, consider you have a proteasome. A proteasome looks like a trash bin. So with a trash bin or a cylinder with two openings on each side. This trash bin likes to be either in one orientation, put to the side, or into the upward orientation, looking along the electron beam, but not in an orientation in between. That means we do not get all the kind of orientations, and by not getting all the orientations, that means not all the projections. You get also not get the full cannot do the construction fully. We miss parts here in the Fourier space, and the back transformation is incomplete. So here again, what is done here. We have this initial images, we have this model projections, we have the reconstruction, initial model, final structure, I think what I mentioned. So, going back to this problem, yes, what this means. Look at this one here. That is your image, that is your object. You do now, let's say, single particle analysis of this strange object. It looks like me falling from the tree. Okay, so you collect a few thousand of these kind of particles, which look like that. But this particle likes not to adopt all kind of orientations. So you see here, you only get a certain kind of degree. You would require the full kind of orientation, all slices around. But you see here, there are some missing because it actually does not orient itself properly in all directions. Nevertheless, you can do the back Fourier transformation, you can do the reconstruction, and that's how it looks like. So before, it was looking quite optimistic, yes, After that, yes, it looks a bit pessimistic, yes, and I would say, yeah, also the brain is bulging out of the skull here, yes, so the structure is distorted, So that is a missing current problem that we do have in single particle analysis and also in electron microscopy tomography. Here another example, what happens when you do not get the full kind of data recorded. Here look at this view of Albert Einstein, yes, and that is how it looks like when you are missing some orientations, yes, and do the reconstruction. I think we could still recognize Albert Einstein here, yes, I think on all these images, but they are not perfect, yes, and clearly, okay, there are some compromises which looks better and not so good. This one looks actually quite okay. That would be like, let's say, the minimal data space you would have to cover to get a more or less complete model of him and his hair and his forest on the forehead, which you do not see here, where actually everything is a bit blurred up. Okay, let's go to the final point for today, tomography. What is tomography? Tomography is pretty simple here. You see here a hair, here's a projector. Let's say this is our object, this is the electron beam, yes, that is our detector. And now what we are doing now, okay, we are on the detector, we collect this kind of projection images, yes. But now the hair is rotating, yes, rotating around this single axis here, perpendicular to the electron beam, and record this kind of different kind of projections we get out from this hair. So that's called tomography. Here you see the idea. First you have the object, you rotate it in the incoming beam, you get different kind of projections, yes, And then at the end you do the back projection in silico, that means in the computer, to reconstruct the object from before. For this kind of technique, you need a sample holder that can be rotated in the electron microscope so that you can do a tomogram, A tomogram is a series of this kind of images, a series of images, you know, to do the projection. That will overcome you, for example, the problem, you know, we have mentioned before, for example, in certain cases where you do not get all the kind of orientations of a given particle. However, please be reminded, you know, when you have a rotatable sample holder in the electron microscope, you can rotate it from -60 to +60 degrees, or maybe -70 to +70, but not more, because otherwise it would collide with the electron beam itself. So you cannot get the full rotational range anyway by tomography. So you have a missing cone problem in any case. So here you see some from our earlier works with the Baumeister group, that I think is still 20 years ago, from some chaperone. It looks pretty similar, you know. Here you see what actually the original images looked like, you know. It's really very blurry, yes, you see very poor contrast, you know. At that time, you know, there was a very low resolution structure, I think 25, 26 angstrom resolution, but it could make its point, you know. That was to be done at that time, you know. So you see here, for example, the symmetry of the particles, an 8-fold symmetric complex that is coming up here. And this one could be then filled up by the jigsaw strategy with some high resolution structures to explain what's going on, you know, in terms of the mechanism of this. It's closing up here a region and opening up and closing up here a region, you know, here on the very top by these kind of helical elements by rotating a domain by about 70 degrees. That's a simple mechanism, you know, that we found out by this technique. So, you know, electron microscopes are interesting because you can also do, and especially tomography is very interesting, because you can also do some really fancy experiments. So that was what we mentioned before, single particle analysis, yes, you know, as we have seen before, you know, with tilting or without tilting, yes, depending whether you have enough orientations anyway available, yes, you know. But you can, of course, also use cells, yes, or bacteria to immobilize them on the grid, at least when they're not thicker than 1 micrometer, and do the tomograms. You know, so that means you can record structures of much, much more complex biological situations. Here on the right-hand side, you see here some reconstruction of a Golgi apparatus, of some part of the Golgi apparatus, yes. Very fancy. I think I have it more fancier here, you know, also with some reconstruction. So you see here a reconstruction of this kind of Golgi apparatus in its context with transport vesicles and so on. Ribosomes which are attached, you know, probably ribosomes, I don't know what these kinds of dots here are, you know, attached to the surface of ER-like structures. So you can make pretty cool pictures and 3D reconstructions of biological objects. Please remember, that was 2001. Now this is now mostly routine-like work in a well-equipped EM laboratory. So what can be done nowadays in 2018? So that's an interesting example. I mentioned the proteasome. The proteasome is actually the trash bin of eukaryotic life, eukaryotic cells. It's degrading proteins. So it exists apparently, you know, the 26S proteasome large complex exists in 2 different states. One is the ground state, where it's mostly resting, and one is the active state, where it's highly active and actually processing ubiquitin-related proteins that go for degradation. So these can be discriminated already here at pretty low resolution by electron microscopy, but one can now do is to study cells under different kind of cellular stress, you know, or corresponding cellular conditions, and quantify now all the proteasomes which are in the single cell, in what state they actually are at the moment. Here, ground state, yes, in green, and substrate processing, that means degrading state, in blue. And that is pretty cool because you can do this on a single cell level. You can do real statistics on this. You can also learn something about the localization about it. Please remember, of course, you know, when you do the electron microgram, they are not green and blue, yes, you know, they have been discovered by the software that here, these kind of volumes, which are colored green, correspond to volumes which correspond to the ground state, and those ones colored blue here correspond to the volumes of the substrate processing state. So one can now try to get also large macromolecular complexes in situ in the cellular condition. What's also cool about cryo-EM nowadays is one of the problems that we do have in X-ray crystallography, also in some part maybe also in NMR spectroscopy, is mostly we see a single conformation. So that means assume like here, for example, here we have two very different conformations for one and the same complex. But when we crystallize something, it will either fall into this one or that one, yes, because it's forming a crystal. But that's not reality. In reality, we have ensemble of different conformations for many kind of protein complexes or some enzymes. And by the electron microscopy method in general, we map them all at the same time. We have the problem of doing the reconstruction and splitting them into the kind of different states. But that is something that depends on data quality, data number, and data processing software, and can be overcome now. So one can even get some idea about the conformational heterogeneity. So that's the reason, why these former disadvantages have been done most to previous disadvantages. For example, missing cone effects, are known, but there are new techniques that have been developed, like subtomogram averaging or sample holders, where you can tilt around two different axes which are perpendicular to each other, or other methods, yes. And also, it's a pretty, one of the disadvantages in the former time was it requires much more computing time, than X-ray crystallography, the time for doing that, but, this methodology is increasingly standardized, and computers are not anymore the limiting factors. So computing speed is not anymore the limiting problem that we have here with this very tremendous image reconstruction properties. One of the advantages is you need only small materials of material. No crystallization is required, which is very good. And the particle size, it is not maybe everything what you like. So for example, when you have a protein of about 25 kilodalton size, that is too small for cryo-EM because it would not fill more than 1 or 2 pixels of the detector. So clearly you need to have something like an image, yes, and that image is determined by how many pixels you can occupy in its projection on the detector. But techniques nowadays can go down up to about 80 kilodalton, that is 60 kilodalton. We have seen it for hemoglobin, so it's getting down quite a bit, and I think we can expect some further improvement in future. So I think most structures you find now in literature are still in the pretty large region, 100 kilodalton or larger. Many of them are also membrane proteins which cannot be crystallized, which are only accessible by cryo-electron microscopy, and the resolutions we can keep with cryo-electron microscopy, they are going below 3 angstrom now for many cases. The resolution definition of resolution is also a bit different, in electron microscopy than in X-ray crystallography, because it's a non-uniform resolution that you do have because of the reconstruction in cryo-EM. But there are examples where you even have structures nowadays made with even less than 2 angstrom resolution, and that is really cool because it means you could even study enzyme substrate complexes and resolve the substrates in these kind of samples. So and with this, I guess I'm at the end. Maybe just to remind you, the electron microscopic structures are all deposited in the RCSB protein database, which I have mentioned to you in one of the former exercises on Thursdays. But there's an additional database, the EM data bank, yes, that has a unified data resource for all the kind of EM structures, also the kind of reconstructed maps, by which the structures have been made, and that can be also being gone through. And the number here is an old number. Let's make a look how the number now in 2020 is. Here, 1,759 released entries. What is the number now today? So that is an interesting question, I want now to answer. So let me just think here. EM database. EM database. How many structures do we have now? Let's make a look. How many structures do we have now? There's some number here. 10,945 released maps, 3,674 associated publications. So you see, in these 7 years, we have now more than sevenfold as many entries as we had, in 2013. That means, the number of structures is rapidly going up because the wet labs, the laboratories, the biochemical laboratories have many samples in the past accumulated that could not be solved neither by X-ray crystallography nor by NMR spectroscopy and have been left to cryo-EM. So let's see what the future gets. Thanks a lot for your attention.